Let's get going. I'd like to welcome everybody to our first digital presentation event following hot on the heels of the AGM a couple of weeks ago. I'll hand over now to Andrew Crosby and Emma Baple, who are going to do their presentation, HSP, a Eureka moment.
So we've been working on HSP since really I became an academic some 22 years ago at St George's Hospital in London. And the reason HSP has always been of interest to me personally is because of the specific nature of the condition. With its often later onset and progressive nature, it's entirely the kind of condition which it should be possible to intervene and prevent or delay the onset of those symptoms and those difficulties. And we called the talk today a Eureka moment for reasons that will hopefully become clear a little later on, as will the relevance of the picture of the bath. So as we work our way through the talk, both Emma and I will talk around each of the slides, and this one's for Emma.
So we called this slide Hereditary Spastic Paraplegia, a Clinical Conundrum. And the reason that we called it this is because HSPs are a very variable group of neurological conditions. So if a patient comes into the clinic and has a diagnosis of HSP, all that really tells us is that they have this common feature, which is this progressive lower limb stiffness and weakness. But it doesn't tell us about the underlying cause of their hereditary spastic paraplegia, or whether they have what we call a pure form of hereditary spastic paraplegia, where they simply have the progressive lower limb stiffness and weakness, or whether it is what we call a complex form of hereditary spastic paraplegia, where they may have other features such as problems with learning or seizures. So what it does tell us, though, is that each of those people have a problem with what we call the upper motor neurons. And I'm going to move over to this diagram on the right hand side and just explain a little bit about what happens with those upper motor neurons and why they're important. First, I'm going to talk about the lower motor neurons. So the lower motor neurons effectively connect our spinal cord to our muscles and pass messages from the spinal cord to the muscles. But without the upper motor neurons, we aren't able to tell those muscles what to do. So if you want to kick a ball or you want to lift up a glass of water, you think about that in your brain and you need to get that message to your muscles. And that message goes through the upper motor neurons, and that it goes from the brain through the upper motor neurons to the spinal cord, and then smooths out that message going from the lower motor neurons to the muscles. So we're able to coordinate this very complex movement that we need to do with our muscles if we kick a ball or lift a glass of water. And people who have HSP all have a problem with the way this upper motor neuron functions, and over time it fails to function as it should. So given that we know that everyone who has a diagnosis of HSP has a problem with the upper motor neurons, investigating all of the different types of HSP together and looking for the underlying problems that are going on in those motor neurons and the molecular commonalities inside those motor neuron cells will help us to understand all the different forms of HSP. Now, there are more than 80 genes that have been associated with HSP over the years, and some of them cause more than one form of HSP. Every time we learn about a new gene that's associated with HSP and the function of that gene inside cells, it provides knowledge to help us with studying other forms of HSP.
So this slide is going to tell us a little bit more about the many genetic causes of HSP and the genes that have been associated with HSP. But before saying a little about them, it's probably best just to remind us all what a gene actually is. So we have 22 or so thousand genes in our DNA, which together form an instruction manual. They tell our bodies how to make and grow a human. And each of those genes is important for the way in which different parts of our bodies develop or function, and many of those genes are important for the way that the upper motor neurons develop and function. And some of those genes have been found to have spelling changes in them, and those spelling changes in those particular genes that are important to the way the upper motor neuron functions can cause HSP, have been associated with HSP. But the problem is that for most of those 22,000 genes in our DNA, in our genome, we don't really have a very good scientific understanding of what their actual very specific molecular roles is. And so because of that, we've grouped the many HSP genes together very broadly, under very broad functional umbrellas, if you like, which tell us somewhat a little bit about what those genes do, but still without knowledge of the specific roles of each of those genes. And so because of that, we don't really have clear knowledge about what any common root molecular cause of HSP, what the core molecular common problem may be that actually leads to HSP. So in Exeter, we've placed a great deal of emphasis on trying to find the genes that, when they have a spelling change, they can be associated with HSP. And through those studies over many years, we've been fortunate enough now to have found 15 genes that can be associated with HSP, with more, we think, in the pipeline. But actually, finding a gene that can cause a condition is only really the very beginning. A huge amount of very complex, time-consuming and often quite expensive work is then required to understand what the role of the molecule is that that gene encodes. So as well as trying to discover more about the function of each of these genes, these HSP genes individually, we've also placed a great deal of effort in a different approach, really, which is to look for things that they have in common. We know that each of these genes causes a problem with the upper motor neuron, causes the upper motor neurons not to work imperfectly. So we believe that there will be common abnormalities associated with the molecular outcomes of each of these genes. And so we've been developing new ways and new tools to try to look at different genetic forms of HSP in parallel to try to find what these commonalities may be. And in particular, we've placed a great deal of emphasis on investigating a specific biochemical process that we believe is common to multiple types of HSP, which is highlighted by one of our genetic studies, which we'll tell you more a bit later about, and our Eureka moment.
So this family, who Andrew saw back when he was working at St George's Hospital and University in 1999, really highlights the problem we face in the clinical genetics clinic when we are trying to help families with hereditary spastic paraplegia. So people are referred to us either to identify the genetic cause of their hereditary spastic paraplegia, or because they would like a test to determine whether they may develop HSP going forwards, because we've identified the cause in another member of their family. But what they really want to know when they come for that test is when they might develop symptoms, how they might be affected, and how that condition might progress. Unfortunately, even if I can offer them a test, I'm really not very often able to give them that information. And this family highlights why that is. So this lady here was the first person in the family that Andrew met, and she had upper and lower motor neuron clinical signs and had been diagnosed with motor neuron disease. Her daughter, who was aged 48, had no signs of hereditary spastic paraplegia or motor neuron disease. Her sister had been given a diagnosis of hereditary spastic paraplegia. And her son had been given a diagnosis of a particular form of hereditary spastic paraplegia called Silver syndrome, where you also get hand muscle wasting, when he was only 18 years of age.
So through working with families in the UK with HSP and through looking at the literature to try to learn more about what had been discovered and what was known about HSP, which at that time wasn't very much, we became aware of a number of historical studies that had been undertaken in the United States, which had described forms of HSP which were present in the Amish community. Unfortunately, at that time, the studies being undertaken in the 1960s, very little was known about the nature of those conditions. All that could be done really at that time by the clinical teams was to describe the features that they saw in patients with those types of HSP, but nothing was known about the genetic causes, nothing was known about the molecular basis of those conditions, so no diagnostics was available, and families really had no counselling or appropriate care that could really help them. So to try to help with this, and also to try to learn more about HSP more broadly, we contacted the clinical teams who cared for those families to see if they'd be interested in undertaking genetic studies to try to understand more about what the root genetic causes of those forms of HSP were. So we set up in around 2000 a program to begin doing that work. And one of the benefits of studying HSP that we found in the Amish community, which was an amazing privilege to be part of this work, was to learn more about their community and their nature, which as part of that they keep these wonderful genealogical records, very extensive genealogical records, which link together families and family members over many generations. And so we're able to put together these huge family trees, which you can see in the bottom right hand side of this slide, which shows how the various families link together. And in genetic terms, because we're not very good at drawing, we represent people as squares and circles in these diagrams, and that's what these pedigrees tell us, these drawings tell us. The blackened symbols that you see at the bottom of this pedigree are the people who had HSP that we studied back in 2000. And through that work, we identified 5 different genetic causes of HSP, different types, genetic forms of HSP present in the community. And that's because the genetic studies that we could undertake were so powerful because of these genealogical records that the Amish kept.
So we've talked a lot about genes and finding genes, because that's where Andrew and I both come from. We're both geneticists. But why is finding genes important? Well, it's finding genes is important because it enables us to offer people a genetic test to determine whether they're at risk of developing a particular condition like HSP. But really, as Andrew mentioned earlier, it's only the first step in the process. So identifying the gene then allows us to look and see what that gene, what that message is doing within the cell. And at the time that we identified the genes that cause HSP, for the majority of them, the function was really either unknown or very poorly understood. And so we've had to undertake painstaking and really very time-consuming cellular studies and experiments to try to discover the function of each of these genes. Sometimes, though, as Andrew mentioned, the function of a gene is really well known, and it's then that you might get a breakthrough, and it's that breakthrough that we want to come on and tell you about now.
So, as we mentioned earlier, we have around 22,000 or so genes in our DNA, in our genome, and each of those genes encodes for, it makes a particular molecule, a particular protein. And so inside each of the cells of our body, and there's a diagram of a very basic representation of a cell on the right hand side of this slide. Each of those thousands of molecules work together in each of our cells in concert to undertake particular and very specific roles inside our cells or bodies. Some of these molecules are important for the structure of a cell. They're part of the scaffolding or part of the skin of that cell. Some of them are important for producing energy and heat, and in particular there are what we call metabolic processes which are important for the production of energy and heat, which involve fat processing pathways, so the way that fats are altered and digested and used inside each of our cells. So, as Emma mentioned earlier, unfortunately most of the genes in our DNA, in our genome, we don't really have a very clear understanding of what the gene's function actually is. But for a small number of genes, we do have a very clear understanding already from studies that have been undertaken historically about what the molecular role of that gene is. And this was the case for one of the genes that we discovered that can lead to HSP when spelling changes occur in that gene. The gene is called SPG5, and that gene had a very well-known specific role in one of these fat processing pathways. The molecule didn't do anything else, it just had this very one simple job in altering the way fats are processed inside the cell, and it's a very specific and particular fat processing pathway that this molecule is involved in. And that was really important because that told us that that fat processing pathway, that specific pathway, is really important for the way that upper motor neurons work, and that disruptions in that pathway can cause HSP. And so this really was a Eureka moment, and Emma likes to joke with me about this because I actually was physically in the bath thinking about all of this when it actually happened. So it really was a genuine Eureka moment, and in light of that, with that realization, we were then able to look again at what was known about the other molecules and the other genes that cause HSP and see if they may relate to this same or similar fat processing pathways. So now, with the knowledge of this fat processing pathway that we'd learned because of our finding of SPG5, we were able to look again at the literature around the other genes that were known to be associated with HSP. So while there was only basic information about many of those genes, that basic information was enough for us to see that very likely, from understanding the way the SPG5 pathway works, that many of those genes are probably important for that pathway or for similar pathways that work alongside SPG5.
So HSP is what we call a motor neuron degenerative disorder because the upper motor neurons degenerate over time, and it's that which causes the symptoms that a patient with HSP has. But there are other forms of motor neuron degenerative disorder, for example Parkinsonism, spinal muscular atrophy, motor neuronopathies, and although each of those forms of motor neuron degenerative disorder have their own distinct genetic causes, and people who have one or the other of those conditions are not at risk of developing the other condition, what we do know by understanding the genetic basis of these disorders is that the pathways within the motor neuron itself that go wrong in these different disorders have commonalities, and so the more that we learn about one form, the more that we learn about the others. And there are these key biological pathways that are common to the many different forms that we are particularly interested in, because if we can interfere with those and repair them in some way, then we may be able to help not only patients with HSP, but patients with these other group of disorders as well.
So we were able to bring all of these thoughts and this data in the published literature together into a paper that we published earlier this year in April in a wonderful medical scientific journal called Brain. so that all of those thoughts and what we would call a hypothesis is available to the wider scientific community.
So the work that we've done on this fat processing pathway, that we believe when it's affected may be an underlying linking cause of many different forms of HSP as well as other motor neuron degenerative disorders, got a lot of national and international press attention. So it was a leading article on BBC News Health, and we also did a radio interview with BBC4 Inside Science talking about this work. There's also a nice video on the independent website describing the work that was undertaken in our study and also talking about that Eureka moment.
We're enormously grateful to the HSP support group for their funding, which has contributed to Olivia Rickman's PhD studies. And over recent months, and through Olivia's work and the work alongside her, we've been gathering data and information to show that our hypothesis is likely correct. And this work has involved two main areas really. The first at the top of this slide is preparing cell models. So we're using modern gene editing techniques, which probably everybody has heard about, which means we're able to, inside any particular cell, deactivate a particular gene. And we've used this technique, these tools, to deactivate many of the genes that we know spelling mistakes can occur in that cause HSP. So we've been developing cell models, these brain cell models, in the lab that we can grow and culture and study to look at these fat processing pathways inside them. And that's what the bottom part of this slide really is about. We've had to develop entirely new methods to look at these fat processing pathways, and it's taken quite some time and some effort, but we've got there eventually, and we're now able to very, very carefully and specifically analyze these fat processing pathways inside cells to see how the different, if and how the different HSP genes affect the fat processing pathway itself. So now looking forward, and again with the wonderful support of the HSP support group, our aim for the future now is to bring together the work that Olivia's been doing and the other workers in the team to better improve the methods that we've developed, so that we can really consolidate, really show that these understand how these fat processing pathways are affected in HSP. And that's really important because it will enable us to take a completely different approach towards testing. At the moment, because little has been known about the underlying molecular abnormality of HSP, the way in which we counsel families has been through doing individual genetic tests for a particular spelling change that is present in a family, to see how that's been transmitted through the family, and if a person therefore may be at risk of developing that condition. And Emma will talk more about this in a moment. But understanding the biological mechanism, if you like, of understanding the processing pathways that are involved by many different genes, means that rather than looking for individual genes themselves and individual spelling mistakes, we're able to look much more generically at these fat processing pathways, which are applicable to many different types of HSP. And our ultimate hope is to be able to do this from a standard 10 ml sample, the type of test you would have in the doctor's clinic ordinarily when they take a small blood sample from you.
So as Andrew mentioned, we now want to take this new knowledge that we have about this fat processing pathway and the new techniques that we've designed to be able to look in blood and in cells at the abnormalities that we see in that fat processing pathway and refine those to produce a blood test that we can use to help patients. But why do we want to do that? What use is it, and how will we be using it for patients? Well, genetic testing is a wonderful thing. It's amazing how much we've learned about the genomic code in the last few years. And we can tell patients an awful lot now that we couldn't even 3, 4, 5 years ago. But genetic testing is still imperfect. It's still extremely difficult for doctors and scientists to interpret genetic testing results. And that's because there are millions and millions of letters in the genetic code. And we all actually have some spelling changes in our genetic code. And it's those spelling changes that make us all different from each other, that make us all unique and special. And it can be very difficult to tell the difference between a spelling change that is just part of normal human variation, that makes us special and different, from a spelling change that affects the way that the body reads that gene. And the way that that gene message is used inside a cell and tells the cell what to do. So if we were able to do a blood test and look at the abnormalities in a fat processing pathway in a patient with HSP, we can circumvent those difficulties and problems with interpreting genetic testing by essentially cutting to the chase and getting to what we know is that problem. We can see that unique signature, that unique abnormality in the fat processing pathway in the blood. So a genetic test is one thing. Having a genetic diagnosis can be life-changing for families. It means that we're able to offer a test to other members of the family. It gives an answer, a name for a condition, but it doesn't fundamentally change anything about that condition for that person or that family. It doesn't change the fact that currently we don't have an effective treatment for HSP that halts the disease progression or stops it from occurring in the first place. And now that we're able to recognise this abnormality in fat processing in the cells of patients with HSP, that is a very powerful tool. It enables us to look for new treatments that might impact that fat processing pathway, that might stop that abnormality, might intervene with it in some way. So once we've identified potential treatments that might help with HSP, the next step is to trial them on patients with HSP to see if they really do work, and that's called a clinical trial. And this is where the blood test that we've been talking about really comes into its own. This is where we believe the real utility is. What we believe is if you give a treatment that works to a patient with HSP, that lipid signature, that lipid abnormality signature that we see in patients' bloods, we believe we'll see improve. It will change, it will get better. And that's really helpful, because if you take a disorder like HSP, which, as you will all know, is very slowly progressive, and you try to see if a patient is improving when they're given a particular treatment by examining them clinically, it can be really difficult actually to know whether the treatment's getting better or not, or whether it just happens to be that particular day the patient's feeling better or is less tired. But if you can take a marker that isn't affected in that way, that's changing in real time, a biomarker we call it, then that is a really powerful tool for monitoring response to a treatment. And that's where we really believe this could be a game-changing innovation, really, for looking for new treatments for HSP. So thank you very much for listening to Andrew and I today. We wanted to leave plenty of time for you to be able to ask us questions, and please feel free to ask us anything that you'd like. We're here to answer your questions, but we're also very interested to hear what you have to say about our work and the ways that you hope that we might be able to help you. So thank you very much for listening.